Hello and welcome to the Deep Dive. Today we're embarking on a journey to understand the most fundamental force powering all life on Earth. Energy.
Right.
I mean, think about the incredible feat of something like a skunk cabbage actually melting its way through snow and ice in winter,
Yeah, that's amazing.
keeping itself at a cozy 22 Celsius, that's 72 Fahrenheit, just by burning energy.
Yeah.
How does it even manage that? And, what does it tell us about what's happening inside every living cell?
It's a fantastic example, really. It showcases the constant energy transformations that are life, essentially. So for this deep dive, we're digging into a key chapter from Raven Biology of Plants. We're focusing on how energy flows, how it changes form, and how living systems manage it.
Okay.
Our mission really is to pull out the essential insights, make these complex ideas clear without you needing the textbook right there.
Absolutely. We'll be unraveling the universe's basic energy rules, the laws of thermodynamics. Then we'll dive into how cells handle electrons.
The electron dance.
Exactly. Exploring enzymes and then understanding that universal energy money, the stuff that keeps everything running. So, yeah, get ready to unpack some seriously cool biology.
Let's do it.
Okay, let's start big picture. Our planet is fundamentally solar powered. It's bathed in this huge amount of energy from the sun, something like 13 times 10 to the 23 calories a year. Just immense.
Unbelievable numbers.
But here's the kicker. Less than 1%. Less than 1% of that is actually captured by plants and other photosynthetic organisms.
Just a tiny sliver.
And that tiny fraction drives almost everything. It transforms light into chemical, electrical, mechanical energy. It's really the essence of life.
It is. And you could even view evolution as this ongoing competition for using those energy resources most efficiently.
Interesting way to put it.
And to really understand this flow, we first need to define energy itself. Simply put, it's just the capacity to do work.
Okay.
Our understanding has evolved, though. For centuries, people thought heat was some kind of separate, weightless fluid called caloric.
Caloric?
Yeah. But then the steam engine came along, and it forced us to connect heat and motion to work. That's really where thermodynamics, the science of energy transformations, began.
Okay, so let's get into those rules. The first law of thermodynamics. This one seems, well, pretty straightforward.
It is, fundamentally.
Energy can be changed from one form to another, but it can't be created or destroyed. Like an engine. Chemical energy in fuel becomes heat, then movement.
Right.
Some energy gets lost, maybe as friction or heat in the exhaust, but the total amount of energy stays the same. It's like cosmic bookkeeping.
That's a great way to think about it. A bookkeeping rule for energy, income, and spending. And when we talk about a system that could be anything, a cell, a tree, the earth,
right.
and the surroundings is everything outside it, the first law just guarantees the total energy of the system plus its surroundings always balances out after any change.
Potential energy. Kinetic energy.
Exactly. Potential energy is stored energy like oil in a barrel or a boulder perched uphill. Kinetic energy is the energy of motion light, electricity, a moving car. They're always converting back and forth.
Okay, so that's the accounting. But you mentioned the second law of thermodynamics is maybe more relevant biologically.
Yes, much more so. This one tells us about the direction things go, the usability of energy. It's sometimes called time's arrow. It states that in any energy exchange, if the system is closed off, the potential energy of the final state will always be less than the initial state.
Meaning?
Meaning, useful energy tends to get dispersed or lost, usually as heat, becoming unavailable to do useful work. It's why things run down.
Like the boulder rolling downhill, but never back up on its own.
Precisely. Or heat flowing from a hot cup to the cooler air, never the other way around spontaneously. Processes that release usable energy are called exergonic. They can happen spontaneously.
Though not always fast, right?
Not necessarily fast, no. And processes that require an input of energy are endergonic.
Okay, exergonic releases, endergonic requires.
Now, a key factor here is enthalpy, delta H. That's the change in heat content. Exergonic reactions are often exothermic. They give off heat. Like burning glucose releases a lot of heat, about negative 673 kilocalories per mole.
That negative sign means heat is released.
Exactly. But enthalpy isn't the whole story. There's also entropy, delta S. That's a measure of disorder or randomness.
Disorder.
Yeah. Think about ice melting into water, then evaporating into steam. That actually absorbs heat. It's endothermic.
Right, you need heat to melt ice.
But it happens spontaneously because the disorder increases massively. Water molecules are more disordered than ice, and steam is way more disordered than water, more randomness.
Like papers getting scattered on a desk, they don't organize themselves?
Perfect analogy. Organizing them takes energy input. The universe tends towards more disorder, higher entropy.
So heat and disorder, how do they combine?
That's where free energy change, delta G, comes in. It considers both the heat change and the entropy change, factored by temperature. The equation is GDHE.
Okay.
And the crucial point is, for any process to occur spontaneously, naturally, a G must be negative. It has to release free energy that can actually do work. These are the exergonic processes.
So a negative EG means go.
Essentially, yes. And this has huge implications for life. Living things are incredibly ordered. They have low entropy compared to their surrounding.
Right, we're definitely not random piles of molecules.
Not at all. We maintain this high degree of order by constantly expending energy. We're fighting entropy, basically.
Yeah.
Because Earth isn't a closed system like the whole universe might be. It's an open system. Think of it like a fishbowl. Energy and matter constantly flow in and out.
Okay, I can picture that. Sunlight comes in.
Exactly. Sunlight is the key energy input. Photosynthetic organisms capture that light energy. They use it to build complex, ordered molecules like sugars from simple, disordered ones like CO2 and water.
So they store the sun's energy in chemical bonds.
Precisely. They create order locally, powered by that external energy source. Life exists in this temporary, localized defiance of the universal trend towards disorder.
Wow. So life is constantly battling chaos, fueled by sunlight. That's pretty profound.
It really is. And the way this energy gets moved around chemically is often through reactions involving electron transfers.
The oxidation-reduction reactions, redox for short.
Yes, redox, absolutely fundamental. Oxidation is simply the loss of an electron. The thing that loses it is oxidized. Oxygen is a common player because it strongly attracts electrons, hence oxidation.
Makes sense.
And reduction.
Reduction is the gain of an electron, and they always happen together. If one thing loses an electron, something else has to gain it.
A paradeal.
Always. And in biology, often an electron moves along with a proton, which is just a hydrogen atom without its electron. So losing hydrogen atoms is often oxidation.
And gaining hydrogen atoms is reduction, like in respiration and photosynthesis. Exactly. In cellular respiration, glucose gets oxidized. It loses hydrogens to oxygen, which gets reduced to water. That whole process releases energy. It's exergonic. About 686 kilocalories per mole.
Okay.
Photosynthesis is basically the reverse. Water gets oxidized to oxygen, and CO2 gets reduced to sugars using those hydrogens and electrons. And that requires energy input. It's endergonic. It stores that same 686 kilocalories.
So it's a cycle of energy release and storage driven by electron movement.
Perfectly put. But a critical point is that cells can't just release all 686 kilocalories from glucose at once.
Why not? Too much heat.
Way too much. It would be like an explosion, incinerating the cell. Life needs controlled release. Small steps.
Right. So how do cells manage that control?
That's where enzymes come in. And also ATP, the energy currency we mentioned.
Okay, let's talk enzymes. Even those exergonic reactions, the ones that release energy, need a push to get started, right? The energy of activation.
Yes, exactly. Think of it as an energy hill the reactants have to get over before they can roll down to become products. In a lab, you might just add heat to give molecules enough energy to get over the hill.
But cells can't just crank up the heat everywhere.
No, that would damage proteins and membranes. So instead, they use enzymes. These are amazing protein molecules that act as biological catalysts.
Catalysts, they speed things up.
Dramatically. An enzyme lowers that activation energy hill. It does this by briefly binding to the reacting molecules, the substrates.
Okay.
It brings them together in the right orientation, maybe strains their existing bonds a bit, making it much easier for the reaction to happen. And the enzyme itself isn't used up. It pops off at the end, ready for the next reaction.
The marriage broker analogy from the tag.
Exactly. It facilitates the match, but isn't part of the final union. And they are incredibly efficient. One enzyme molecule can handle thousands, even tens of thousands, of substrate molecules per second.
Wow. And they're specific, right? Like sucrose only works on sucrose.
Highly specific. Almost all enzymes are large globular proteins folded into a very precise 3D shape. This shape creates a little groove or pocket called the active site.
The business end.
That's right. And the active site has just the right shape and chemical properties, patches of positive or negative charge, water-repelling areas to bind its specific substrate. So it's a lock and key, or maybe even a glove fitting onto a hand.
Like that example of hexokinase closing around glucose.
Yes, a great example of induced fit. The enzyme might even slightly change shape upon binding the substrate to hold it perfectly for the reaction.
Incredible molecular machinery. Do enzymes always work alone?
Not always. Many need help from non-protein components called cofactors. Sometimes these are simple metal ions like magnesium or zinc. They might help stabilize the enzyme shape or directly participate in the reaction, maybe by holding a substrate in place.
Okay, metal ions. What else?
Other cofactors are organic molecules, often called coenzymes. These are frequently involved in redox reactions, acting as electron carriers.
Like NAD plus GORO, nicotinamide adenine dinucleotide. That sounds complicated.
It looks complex, but it's built from familiar bits adenine, ribose sugars, phosphates. The key part is the nicotinamide ring derived from niacin, which is vitamin B3.
Ah, so that's why we need B vitamins.
That's one major reason. NAD plus can accept electrons and a proton to become NADH. It acts as an electron shuttle. And like enzymes, coenzymes are recycled, so the cell doesn't need huge amounts.
Efficient. What about prosthetic groups?
Ah, yes. If a cofactor, whether it's a metal ion or a coenzyme, is tightly and permanently bound to the enzyme, then we call it a prosthetic group.
Got it. So enzymes often work with helpers. How are they organized in the cell, or are they just floating around?
Sometimes, but often they work in organized sequences, like an assembly line. We call these metabolic pathways or biochemical pathways.
Okay, like a production line.
Exactly. Molecule A enters, enzyme 1 converts it to B, enzyme 2 converts B to C, and so on down the line to a final product. Figure 5.9 visually shows this flow.
What's the advantage of doing it that way?
Several advantages. First, you can group the enzymes for a pathway together in a specific part of the cell, like in mitochondria for respiration, efficiency.
Makes sense.
Second, intermediate products don't really build up. As soon as B is made, enzyme 2 grabs it. It keeps things flowing smoothly.
Less mess.
Right. And third, if you have a strongly exergonic step late in the pathway, it can actually help pull the earlier, maybe less favorable reactions along by keeping the concentration of intermediates low.
Maintaining momentum. What are isozymes?
Good question. Sometimes the same reaction needs to happen in different places or under different conditions in the cell. Instead of using the exact same enzyme molecule, the cell might use isozymes. These are different forms of an enzyme, coded by different genes, that catalyze the same reaction but might have slightly different properties, making them better suited for their specific location or role.
Tailored tools for different jobs. Okay, so we have these pathways. How does the cell control them? It can't just have everything running full blast all the time, right?
Absolutely not. Regulation is key. Cells are incredibly efficient. They only make what they need when they need it. Some control is simple availability of the substrate or necessary cofactors. If they run out, the pathway stops.
Supply and demand.
Pretty much. Temperature is also a factor. Enzyme activity generally increases with temperature up to a point. Rates often double for every 10 degrees rise, say between 10 degree and 40 degrees C.
But too hot is bad.
Very bad. Above an optimal temperature, the enzyme's delicate 3D structure starts to break down and denatures. Think of cooking an egg white. The protein unravels and changes permanently. Denatured enzymes lose their activity rapidly. Figure 510 graphs this peak activity.
Okay, so temperature matters. What about pH acidity?
Also critical, pH affects the electrical charges on the amino acids in the enzyme. Changing those charges can alter the enzyme shape, especially at the active site, and its ability to bind the substrate. Most enzymes have an optimal pH range where they work best.
So cells need to control their internal pH too.
Definitely. But beyond these general factors, cells have more specific on-off switches for pathways. Usually there's one key regulatory enzyme in a pathway, often the one catalyzing the first committed step or the slowest step.
The bottleneck.
Exactly, the rate-limiting step. Controlling that enzyme controls the whole pathway's output. And many of these regulatory enzymes are allosteric.
Allosteric.
Oh, their shape. Right. Allosteric enzymes have the regular active site for the substrate, plus another separate site called an effector site or regulatory site.
Okay.
Molecules called effectors can bind to this other site. This binding causes a change in the enzyme's overall shape, which in turn affects the active site's ability to bind the substrate. It can either activate or inhibit the enzyme.
A remote control switch.
A great analogy. And a really common type of allosteric control is feedback inhibition, also called end product inhibition. Figure 511 illustrates this.
How does that work?
Simple and elegant. The final product of the metabolic pathway acts as an inhibitor for the regulatory enzyme, usually the first enzyme in that same pathway.
So when you have enough product,
the product itself goes back and binds to the allosteric site on that first enzyme. changing its shape and slowing down or stopping its activity.
Shutting off the production line because the warehouse is full.
Exactly. It prevents the cell from wasting energy and resources making something it doesn't currently need. When levels of the product drop because the cell uses it up, it detaches from the enzyme and the pathway starts up again. It's self-regulating.
Very clever. Okay, we've mentioned energy release and control. Let's get to the money. ATP, adenosine triphosphate.
The universal energy currency, yes. So much of what cells do, especially building things or moving things, runs on ATP.
What is it chemically?
It's structurally related to NAD plus RNA. It has adenine, the sugar ribose, but then it has 3 phosphate groups linked together. Figure 512 shows this structure.
Triphosphate. Okay.
And the key is in the bonds linking those last 2 phosphate groups. They're called phosphoanhydride bonds, and they store a significant amount of free energy.
Why so much energy?
Partly because those phosphate groups are all negatively charged and they're packed closely together. They repel each other strongly, like compressing springs. So breaking off that terminal phosphate group releases that stored energy.
How much energy?
When ATP is hydrolyzed, broken down with water into ADP, adenosine diphosphate, and a free phosphate ion pi, it releases a good chunk of energy. Under typical cellular conditions, it's often around 12 to 15 kilocalories per mole.
More than the standard value you see sometimes.
Yes, the standard value of 7.3 kilopala is under very specific idealized conditions. Cellular conditions are different, making the actual energy release usually higher.
And this release is exergonic, right? Drives things.
Highly exergonic. Enzymes called AT passes control this hydrolysis. The energy released powers muscle contraction, literally proteins called myosin walking along actin fibers. It powers pumps that move ions across membranes against their concentration gradients. All sorts of cellular work.
Can you break off the second phosphate too?
You can. Breaking ADP down to AMP, adenosine monophosphate, and another phosphate releases a similar amount of energy. But often, that terminal phosphate isn't just released, it's transferred directly onto another molecule.
Phosphorylation.
Exactly. Enzymes called kinases do this. They transfer that high-energy phosphate from ATP onto another molecule, activating or energizing that molecule, making it more reactive for a subsequent step.
So ATP doesn't just release energy, it passes it on directly.
Often, yes. Think about making sucrose, table sugar, from glucose and fructose. That reaction is endergonic. It requires energy input, about 5.5 kilocalories.
So it wouldn't happen on its own?
Nope. But the cell couples this reaction to ATP hydrolysis. Figure 513 shows this coupling. It uses ATP to first phosphorylate glucose and then maybe fructose. These phosphorylated sugars are now higher energy.
Prime for reaction.
Exactly. Then they can react to form sucrose, releasing the phosphate groups. The overall process, combining the ATP hydrolysis, energy release, with the sucrose synthesis, energy requiring, becomes exergonic overall. The cell spends ATP energy to make the sucrose synthesis happen.
So ATP bridges the gap between energy-releasing reactions like breaking down glucose and energy-requiring reactions like building molecules.
That's its crucial role. The ATP-ATP system acts like a rechargeable battery, constantly being charged up by energy-releasing processes like respiration or photosynthesis, and then discharged to power energy-consuming processes. It shuttles energy around the cell.
Wow. Okay, that's quite a journey through cellular energy.
It really is fundamental stuff.
So wrapping up, we've gone from the sun showering energy onto Earth to less than 1% being captured.
Right, driving almost all life.
To the basic laws of thermodynamics, energy changing form but the total staying constant, and the unavoidable increase in disorder, entropy.
And how life fights that entropy by using energy to build and maintain order thanks to Earth being an open system.
Then the electron dance of redox reactions, moving energy around.
Controlled by enzymes, those amazing catalysts that lower activation energy.
Using cofactors and coenzymes like NAD plus log, working in ordered metabolic pathways.
Regulated by feedback inhibition and allosteric control.
And all ultimately powered by ATP, the cell's rechargeable energy currency, coupling energy release to energy use.
You got it. From the sun down to the molecular level, it's all about managing that energy flow.
Thinking back to that skunk cabbage melting snow, it's using these exact principles, breaking down stored food, glucose, releasing energy via respiration, using ATP to generate heat.
Precisely. And your own cells are doing it constantly, building proteins, sending nerve signals, contracting muscles, all dependent on this continuous controlled energy transformation. It highlights the sheer energetic cost of just being alive.
It really does. It makes you appreciate the constant, invisible work happening inside us all the time. So, here's a thought to leave you with. Every breath you take, every move you make, it's all a demonstration of incredibly efficient energy management. What does reflecting on that sheer energy demand tell you about the requirements, not just for survival, but for truly thriving? Something to mull over.
Definitely food for thought.
Thank you for joining us on this deep dive into the energy of life. Until next time, keep digging, keep learning.